Alright, in this video I am covering a portion of chapter 12, and I wanted to show you how to use what's called the multiplication rule and pedigrees, essentially family trees, for solving problems in genetics and doing genetic analysis. We've already talked about being able to use Punnett squares to predict the outcome of genetic crosses. We did monohybrid crosses, where you're following just a single gene or a single character at a time. We did dihybrid crosses following two genes at a time. And those Punnett squares can be really useful in terms of being able to predict what the offspring of a particular cross may look like. But when you go beyond dihybrid crosses, let's say you wanted to look at 3, 4, 5 genes at a time, the Punnett squares would become very difficult to do because they would just become so large. So I wanted to show you this little trick where we can use what's called the multiplication rule of probability, to make life a little bit easier here. And so the multiplication rule basically says that the probability of two independent events happening at the same time is the multiple of their individual probabilities. So if that's confusing, take for example situation where you're going to flip two coins at the same time, and you want to know what's the probability that they're both going to be heads, land on heads. So of course, for each coin flip, you have a 50-50 chance, right, or one half chance that each coin would land with the head side up. And so it would simply be, 1/2 chance for the first coin, multiplied by 1/2 chance for the second coin. So we'd have a 1/4 chance, that 1 in 4 chance that both coins would land on heads. And so that's the basis of that rule that we're going to use here. So as I just mentioned, what if I was to give you a trihybrid cross like this? So we're following 3 genes, the A gene, the B gene, and the C gene. I give you this cross where we're crossing 2 triple heterozygotes, right? Big A, big B, big C, little c. I give you that cross, and I want to know what's the probability that you are going to get an offspring with this specific genotype. So big A, big B, big C, little c. Now theoretically you could do the Punnett square here, but you are going to have to come up with all the possible combinations of 1A, 1B, and 1C in order to start building that square. And it's going to get confusing and complicated, and as I said, your square is going to be huge. So we're going to use this little shortcut with the multiplication rule. And to do this, all you have to know how to do is be able to carry out a monohybrid cross, which hopefully you can all do by now. So we're going to take this big trihybrid and just break it down into 3 monohybrid crosses. So for example, we're just going to look at the A gene in isolation, where we're crossing a heterozygote big A, little a, with another heterozygote big A, little a. And so we would set that up in a little monohybrid cross here, right? We've got our big A, little a on each side of our square, and we get kind of our traditional 3-to-1 ratio, where we'd get our 1 homozygous dominant, our 2 heterozygotes. All 3 of those would have the dominant phenotype, and then our homozygous recessive. And we're going to do the same thing with the B gene and the C gene, and because for all 3, these are just heterozygotes being crossed with heterozygotes, essentially all 3 squares are going to be the same in this instance, right? So we've got our big B, little b, and so on. And so the way we solve this problem then, remember you're being asked what's the probability that you're going to get an offspring with this particular genotype shown here. So for example, you were being asked big A, big A. Well, if we look at our little Punnett square we did here, basically 1/4, right, would be big A, big A. So we make note of that. Move on to our B's. Big B is what you're being asked about, homozygous dominant. That's going to be same scenario here, right? 1/4. And then you're being asked for the heterozygous big C, little c, which we've got 2 of those over here. So 2/4 of our offspring, or we could reduce that to 1/2, would be heterozygous. So we've got these 3 fractions now, and because this is the multiplication rule, we're simply going to multiply those together. And if you've forgotten how to multiply fractions, you're simply multiplying all the numerators and all the denominators. So 1/4 times 1/4 would be 1/16, and then 16 times 2 is going to be 32, so we'd have 1/32, basically 1 out of every 32 offspring from this trihybrid cross up here should have that particular genotype. So that's really useful. And we could do this same kind of trick on a dihybrid cross as well. It would work as long as you're being asked for a specific genotype, as you are in this question. But I do want you to make sure that you can do a dihybrid cross in a Punnett square as well. And in the assessment that we're going to do online, I'm going to ask you to actually draw out dihybrid and monohybrid Punnett squares, and then probably solve one of these bigger problems with the multiplication rule. All right, so that's multiplication rule. Let's move on and talk about pedigrees for a minute. And if you're not familiar with pedigrees, pedigrees are essentially family trees that are following a specific trait or character through a family. And this is really the main way geneticists study human genetics, because of course you can't cross humans, right, like you would pea plants or other research animals for that matter. But we can analyze existing families and see how traits are inherited there. And of course, the laws of inheritance are the same in humans as they would be in pea plants or in mice or really any other organism that sexually reproduces. So if you haven't looked at a pedigree before, just get you comfortable with the notation here. So females are always represented by circles, males by squares, and we would represent a couple that has children by connecting them with a horizontal line like this, and then lines branching off of that horizontal line to each of their offspring, each of their children. So this couple here had 1, 2, 3, 4 children, and so on. Then we could go on to the next generation. So however many generations of information you have, you could include in one of these pedigrees. But really the point of one of these pedigrees is to try to look at the inheritance of some particular trait, or maybe it's a genetic disease that runs in that family. And you'll notice on this pedigree, some of these individuals are shaded in or darkened in. So those would be the people in this family that actually have the particular trait or disease that we're studying in this instance. The individuals that are white, that are not shaded here, don't have that particular trait. So they're using the term affected or unaffected here. As you'll see in the next image I'm going to show you, sometimes there'll be people that are considered carriers for a particular trait, so they don't actually have the phenotype or have the disease, but they do have a gene that they could potentially pass that on to their children. So sometimes you'll see that represented with half shading like this. So really, if you're looking at one of these pedigrees, the first thing you want to try to determine is whether the particular trait that you're looking at is inherited in a dominant or a recessive fashion. And this, as you can see, is an example of dominant inheritance. How do we know? Well, dominant inheritance is characterized by the fact that the trait does not skip generations. It's basically being passed from parent to child, appearing in every generation. So that doesn't mean every person is going to have that trait. So for example, here we've got our generation 1 parents here. the male has the trait, the female doesn't. And if you look at their children, right, 3 of their kids now have that particular trait, but 1 doesn't. Um, but if we look at the 3 that do, and they all happen to have kids, we can see at least 1 of their kids also has the trait, right, over here, for example. So we're seeing this trait appear parent to child in every generation, like direct descent. So that would indicate dominant pattern. And what that would tell you, if you looked at the pedigree and saw that pattern and said, okay, this is a dominantly inherited trait, that would mean that everybody that's shaded in this pedigree that has that trait has at least 1 dominant allele for the gene that controls that trait. So if I just use the letter A here just randomly to represent the gene controlling whatever the trait is we're looking at here, that means every shaded person is either going to be homozygous dominant big A, or heterozygous big A, little a. Okay? And I just filled in 3 here, but every shaded person would have one of those genotypes, have the dominant genotype essentially. Whereas in recessive inheritance, that's always characterized by these traits skipping generations or skipping multiple generations and then suddenly appearing, which is what we're really seeing here. So in this case, really the only individual that actually has the trait here is this one down here that's in generation 4 that's shaded in. So we had 3 generations where nobody showed the trait. Remember, the people that are half shaded here, they don't have the trait, but they're quote unquote carriers. So 3 generations this family did not have whatever this trait, or let's say it's a disease is, and then all of a sudden it pops up here. So it was skipping at least 1, but in this case 3 generations. And you'll also notice in this case, the reason we had this individual all of a sudden produce this trait is we had some inbreeding in this family. So we basically had first cousins having kids with each other. And so what this means, if we have a recessive pattern, is that every individual with that particular trait is going to be homozygous recessive, so little a here. And if we step this up to these first cousins that were carriers, that means they were heterozygous, so big A, little a, because they have that 1 dominant big A, they don't have the trait, but they are carriers because they have that recessive allele. And because they were having kids with each other, right, this poor individual got the recessive allele from each parent and is now homozygous recessive. So this is why there's laws against discouraging marrying your close relatives, because if you think of this in terms of the larger population, if this is a rare recessive disorder, the chance that one of these carriers in this family marries another carrier just at random from outside the family is very small. So it would be very unlikely that you would end up then with a child with this trait. But because they're marrying within the family, it becomes much more likely. This was very famous, an issue, for example, with hemophilia in the royal families in Europe. So hemophilia is a recessive disorder where your blood doesn't clot, so you could bleed out, have a serious event just from a very small cut. And as I'm sure you're aware, right, at least this is the way it used to be, right, royalty was expected to marry other royalty, so that's a pretty small pool of people to choose from. So you had these royal families across Europe that were basically all pretty closely related to each other. All right, so let's try and work through one of these pedigrees. I'm going to give you a problem like this on the assessment that I'm going to have you do for this section. So let's say we have this pedigree, and you can see there's a number of people that have a particular trait in this family. First thing to ask would be, is this trait dominant or recessive? And what you'll notice is we are seeing the trait skipping generations in this case because, for example, we look here, Arlene has the trait, her daughter Anne does not have the trait, but then Anne's daughter Carla does, so it skipped over Anne in this particular case. And if it skips generations, that indicates a recessive trait. So that would mean if it's recessive, then everyone who's shaded in this family is going to be homozygous recessive. So those will be the easiest individuals to determine their genotypes. So what about some of the other people in the family? Let's see how many we could fill out. Let's go back up here and start with Arlene's husband, George. So George doesn't have the trait, which means he has to have a dominant A allele for this particular gene, right? A dominant version of the particular gene controlling this trait. But George and Arlene did have two homozygous recessive kids. So that means we know that George has to be heterozygous because Tom and Wilma, they had to get one of their little A's from mom, and they had to get the other little A from dad. So that lets us know George's genotype in that case. What about Sam here? Sam didn't get the trait, so that means he should have gotten the big A gene from dad, and of course he would have gotten a little a recessive from mom, so he should be heterozygous just like dad. Anne over here should be the same story. What about Anne's husband Michael? So he's coming in from outside the family. We don't have a whole lot of information about his genetics, but we should be able to deduce based off the fact that Anne and Michael had an affected daughter, Carla, here, who's homozygous recessive, that he also had to be heterozygous. Okay. So in this case, Carla got little a from mom and dad. Then that just leaves us with this side of the family over here. What about Tom's wife, Sandra, and some of their kids? Well, if we look at this, Tom and Sandra, we know Tom's homozygous recessive. They had two kids, neither of whom had the trait. So that means, and we know Sandra doesn't have the trait, so she has to have at least one big A allele, one dominant. But we don't really know, she could be homozygous dominant big A, but she could also be heterozygous. That's just not really clear. And when you can't really say for sure, a lot of times that second allele will just be represented by an underscore like this. And then, of course, we know their kids, Daniel and Ellen, both got a little a from dad, both got a big A from mom since they don't have that particular trait, so they're both going to be heterozygous. Tina's coming in from outside the family there. We can't really nail her down too specifically, but we know she's got at least one dominant allele, and really same deal with their son Christopher. So, it gives you some information on that family. If whatever this trait was that we're following was some serious genetic disease, these kind of analyses could be really useful if you're, a couple in this family trying to decide, what are the chances if we have kids that we might have an affected child with this particular disorder. So genetic counselors would do this kind of analysis and help advise couples in that regard. All right, I'm going to stop there.